Okay, so we're going to be looking at the B2 AQA inheritance section of the syllabus. If we start by looking at a cell, we can see that inside of the cell we have the nucleus. The nucleus stores the genetic information. Now, in the human nucleus, there are 46 chromosomes. Chromosomes are thread-like structures. In the human nucleus, there are 46 chromosomes in total, the diploid number. 23 chromosomes come from mum, and 23 chromosomes come from dad. Now, chromosomes are made up of a chemical called DNA, so deoxyribonucleic acid. Now, sections of the chromosome are called genes. Genes code for our characteristics or proteins. So if we were to look at this, you can see the gene, there is a base sequence, so A, C, T, G, and the order of the bases in DNA code for the order of amino acids. So a different base sequence will lead to a different amino acid sequence and therefore a different protein. Okay. So if we think about it, then there's 23 chromosomes in the human sperm cell, there's 23 chromosomes in the human egg cell, and when the two gametes, the sperm and the egg, fuse together, they create the zygote, which has the diploid number of chromosomes, or 46 chromosomes in total. So if we look at how this then creates our characteristics, we need to go back to the chromosomes. So essentially, there are 23 pairs of chromosomes in the nucleus. Now, the 23rd pair of chromosomes codes for sex. So if you get an XY pair of chromosomes, you will be a man, and if you get an XX, you will be a woman. Now, during meiosis, the sex cells are formed, and one of each of the chromosomes goes in to form the gametes. And it's random as to which chromosomes go from the body cells end up in the gametes. Okay. Now, if this sperm here fertilises this egg, we will end up with a girl. If this sperm fertilises this egg, we will end up with a boy. Okay. The process of fertilisation is completely random. We don't know which sperm is going to fertilise which egg. Now, if we're trying to work out the chance of the offspring being male or female, we can basically do something called a Punnett square. So what we do is we place the male gametes. So remember, male gametes or sperm can either carry an X or a Y chromosome. The female gametes can only carry X chromosomes. That's why it's the sperm that determines the sex of the child, because it's the sperm that carries the Y chromosome. So if this male gamete fuses with this gamete, we get XX, we get a girl. If this egg fuses with this sperm, we get a boy. This egg with this sperm, we get a girl. And this egg with this sperm, we get a boy. So that gives us a 50% chance of a girl or a boy. So if we have a look at homologous chromosomes, remember I said that there are 23 chromosomes in a human cell. One of those chromosomes comes from mum, and one of those chromosomes comes from dad. We call them homologous chromosomes. because you can see each pair are similar. And if we were to take chromosome pair 9, you can see that they have genes at the same loci. So for example, the eye colour gene is in the same position on both the male and the female chromosome. Okay, so just to remind you again, this 23rd pair denotes what sex you are. So we can see these are different, so it's likely that this person is a boy. Coming back to homologous chromosomes, so the chromosomes in a matching pair contain the same type of genes that code for the same characteristic. So like I pointed out in the last slide, you can have a gene for eye colour located at the same place in the homologous chromosome pair. However, they might have a different version. So for example, the female chromosome that you've inherited, or the chromosome you've inherited from your mother, you might have a version for blue eyes, whereas the version you inherited from your father, okay, so your dad's, you might have inherited a version for brown eyes. These different versions of a gene are called alleles. Look a little bit further, we represent alleles using letters. A dominant letter is always a capital letter. Okay, so start with that, find out what the dominant characteristic is, and that gives you the capital letter that you're going to use. So in this case, when we look at the gene for eye colour, we can see that the allele for brown eyes is capital B. So then the recessive allele always has the corresponding small letter. So the allele for blue eyes is recessive, so we give it a little b. Okay. When the allele pair come together, so one from your mother, one from your father, for each characteristic we call it the genotype. So we could have big B, so homozygous dominant. We could have big B, little b, so heterozygous, or we could have a genotype little b, so it's homozygous recessive. So if we talk about this, so the allele pair for each characteristic is called the genotype. The physical expression of the allele pair is called the phenotype. So if we have a look at this for the example of eyes, we can see the genotype is big B. So this is homozygous dominant. Okay, homo meaning the same, dominant meaning you've got two of the same dominant alleles. Heterozygous, think if you're heterosexual, you like the opposite sex, so it's different. So we've got different alleles, so in this case we've got one dominant allele and one recessive allele, or one allele for brown eyes and one allele for blue eyes. And then we've got homozygous recessive, so we've got two similar alleles, or two alleles that are the same, should I say, for blue eyes, and it's recessive. Okay. Now remember, dominant, you only need one copy of the allele to get the feature, whereas with recessive characteristics, you need two. So the phenotype, what we see, the physical expression for this genotype would be brown eyes. For the heterozygous genotype, so big B, little b, because brown eyes is dominant, again it would be brown eyes. And the only way we can get blue eyes is if we get two recessive alleles, and therefore blue would be displayed in the phenotype. So one thing to do in your exam is predict the outcome of a blue-eyed parent mating with a brown-eyed parent and what their offspring would be. Okay, so remembering that the phenotype brown eyes is controlled by a dominant allele, and the phenotype blue eyes is controlled by the recessive allele, or lowercase b. So for blue eyes, the genotype can only be 2 of the recessive alleles, whereas the genotype for brown eyes can either be heterozygous or homozygous dominant. So if we start by saying we have a blue-eyed parent, and we also have a brown-eyed parent. Okay, now this is obviously homozygous, recessive, and in this case, let's say that the brown is heterozygous. Okay, so this is the phenotype of the parent. Okay, so we're timesing those together. So the genotype for the parent, so remember homozygous, recessive, so we've got 2 lowercase b's, and remember we've got heterozygous, so we've got 1 dominant and 1 recessive allele. Okay, so we now want the gametes. Well, the gametes are either for this parent, say this was dad, is either going to carry a lowercase b or a lowercase b, so it's going to carry recessive allele, where the mother's gametes are either going to carry a dominant allele or a recessive allele. So what we can do now is we can place this into a Punnett square to show the offspring. So we've got gamete here, and we've got gamete here. Okay, so remember we said mum was big B, little b, dad was little b. So then we fill in our genetic cross. So we've got a genotype big B, little b, big B, little b, big B, little b. Oh, no, I made a mistake. So that is little b, and little b. So we've got 50% will be blue, and we have 50% that will be brown and heterozygous. And it's important that you lay out your working like this so you can show your thinking. Okay? You might even want to label under here so it shows that you know what the phenotypes are of the offspring. So this bit here is showing the offspring. Okay? So you're expected to know about 2 genetic diseases. One of them is cystic fibrosis, and this is caused by a recessive allele. So remember that means that you need 2 copies of the affected gene. So if the normal allele is big C, then the recessive allele is little c. So you basically need to be that. Now, some of the symptoms of somebody who has cystic fibrosis is basically they produce a thick mucus. that blocks the air passages, and it can cause chest infections. And one of the only ways of treating these chest infections is through antibiotics, or through a lung transplant, or using an inhaler to basically make breathing easier. Now, mucus can also occur in the pancreas, which means you can't release enzymes, which can then cause digestive problems. So again, you could be asked to predict the probability of having cystic fibrosis if both of your parents are carriers. So that means they carry one copy of the defective allele. So again, these are mum's gametes and this is dad's gametes. We can see that there is 25% chance of not being affected, so being homozygous dominant. There is 50% chance of being a carrier, so having one copy of the recessive allele, and there is 25% chance of having cystic fibrosis, so having two defective copies of the allele. So in total, there is 75% chance that you will not have the disease and 25% chance that you will. The second disorder that you need to know about is polydactyly. And it's a genetic disorder of the 7th chromosome that basically results in you having extra fingers or toes. Now, this is caused by a dominant allele. Now, the way I remember that this is dominant and not recessive is because it's got a D in the middle, whereas cystic fibrosis has an R for recessive. So hopefully that will help you remember it. So remember, if it's caused by a dominant allele, we'll give it a capital P to show that just getting one copy of the defective allele will give you the disease. So you just need one abnormal because it's caused by a dominant allele. So if we then have a look at this, so complete the Punnett square to show the probability of having polydactyly if both of your parents do. So they are heterozygous for the disease. So both parents have a genotype, big P, little p, big P, little p. So again, if we fill out our Punnett diagram, remember to label it correctly, we can see we've got 25% that are homozygous dominant, 50% that are heterozygous, and 25% that are homozygous recessive. Now, because you only need one copy of the affected allele to have the disorder, that means that there is a 75% chance that the offspring will have polydactyly. Now, you might be asked, why can you not be a carrier? Well, you can see here, this one and this one, they can't be carriers because if they just get one copy of the allele, even if they've got a recessive allele, that won't make a difference. They will still have the disease. Now, one of the trickier questions that you might get is to do with inheritance of polydactyly in a family, and you could be given something called a pedigree diagram. Now, first of all, you need to get your head around the key. So this basically shows you that the squares represent males and the circles represent females. Now, if they've got a line joining them together, that basically means that they are parents, and any of these lines that come directly off the parents are their offspring. So it tells you that polydactyly is caused by a dominant allele, and it gives you a capital D to represent that, and the recessive allele of the gene is represented by a lowercase d. and it wants you to use one genetic diagram to show the inheritance of the polydactyly gene by R and S. So first you need to work this out. Well, Q, to start with, is the unaffected female. Now if it's unaffected, that must mean that Q is little d, little d. Now P is an affected male. So that basically means he can either be big D, little d, or big D, big D. Now, if you look at the offspring, you can see that half of the offspring have the disease, and you can see that half of the offspring do not have the disease. So you need to think about, well, what would the genetic cross be? So which of these genotypes would the parents have to give that ratio? Well, if P was homozygous and had babies, homozygous dominant, and had babies with Q who was homozygous recessive, so if we do that here, you can basically see that all of their offspring would have the disease. So we know that that's wrong. Okay, so cross it through. So that must mean that P is big D, little d. So the gametes are either carrying a copy of the dominant allele or recessive, and Q, the gametes are carrying recessive alleles. So basically that means that you are going to get something that looks like this. Okay. And so then we've got, so R, because it is an affected male, must be big D, little d, and S, because it's an unaffected female, must be little d, little d. Okay, I know this is a very long video, but this is the last thing I want to talk to you about. So obviously genetic diseases run in families, and now there are genetic tests to see whether or not you are carriers for the allele, and therefore that you could potentially pass it on to your offspring. So there's a couple of things that can happen. Well, to start with, you can basically have IVF, so you can create embryos in a test tube, and then you can test one of the cells from these embryos to see whether or not they have the disease before deciding whether to implant the embryos back into the uterus. Obviously, if the embryo does have the disease, it would be discarded. So there is the ethics of potentially killing lots and lots of lives in the hope to find one that doesn't have the disease. Obviously, another way you could do it is screening of the fetus when it's in the uterus by testing some of the cells, and again then the woman would have to decide whether to abort the fetus should it have the disease. And this is obviously very controversial, and many people are concerned that if we can screen embryos to rid them of genetic disorders, and we can screen fetuses in the womb and abort them if they have genetic disorders, that potentially we could screen for lots of other characteristics. So maybe the eye colour or the hair colour or the sex or the skin colour. There are some countries that terminate when they find out the sex of their child. So there's a degree of infanticide, and we talk about this as designer babies. On the other hand, if you genetically screen an embryo that maybe has cystic fibrosis and are choosing the embryos you're putting back into the uterus to grow, then you, as a parent, you are not allowing your child to grow up with a disease that ultimately is going to lead to them having a very short life. In your exam, you might be asked to look at or evaluate embryo screening. I hope the video has been useful, but I also know that it is very long. If you have any questions, please contact me.